Okay, we can start now.
Hi, everyone. I'm Andrew McCulloch. How's everyone doing? Okay, I'm grateful for you to come along. I don't really have a chalk talk per se. It's more of an iPad talk. How many of you are bioinformatics majors? Maybe give me a thumbs up. Okay. So most of you. So how many are in bioengineering? Okay. What about computer science and engineering? Any from biological sciences? Great. And did I miss anyone? Well, I'm really pleased to talk to you. You know, there's your major. I think if I had a chance to do an undergraduate major today, like my son, who's like going to be a new freshman in the fall here, I wouldn't be choosing mechanical engineering like him. I'd be choosing your major. I'd be choosing bioinformatics major. In fact, I was on the committee that helped design it. And I just thought it was the coolest major of all. So, so I'm really happy to talk to you. I thought what I'd do is I'd give you kind of a high level idea of what bioinformatics and systems biology is to me and where it fits in the context of computational biology. And then I'm not going to talk to you about bioinformatics per se, because you all know about bioinformatics, and for us, it's like a tool that feeds into the rest of the pipeline. But you know, take it for given that we couldn't do any of this without bioinformatics. But I think what bioinformatics and other branches of computational biology have in common is that they're integrative. And I see three main ways in which computation is integrative in biology. And the first way is essentially information integration. And this is what we think of as classical bioinformatics. This is annotating, collecting, annotating, databasing, searching, querying, and doing statistical analysis on the components of biological systems, particularly genomes and proteomes and the networks that they form, and how they relate to biology in a range of different contexts. So it's integrating information from a broad array of different experimental measurements in biology. No, I can't make this advance. Uh-oh. Okay, we go. The next level of integration is really what we call functional integration, where now that we have networks that we can reconstruct into modules and functional subsystems, like what you see here, is a little cartoon of a network in a cardiac muscle cell that's responsible for ionic currents and calcium handling, called the excitation contraction coupling system. So we can use bioinformatic information and experimental information that's well organized, annotated, and interrelated in order to reconstruct modules, networks, and functional subsystems that we can then model. And we can model them using system science by taking the functions of each of the components and building systems models of the entire system or subsystem that then predicts the emergent properties of the system. So this is what I would call functional integration. You take function of components and you predict function of systems And I think this is the sort of, from a fundamental point of view, the core goal and purpose of systems biology, to take the functions of components and predict the functions of systems. Again, it's integrated. But there actually is a third kind of integration, and that is integration across physical scales of biological organization, from molecule to organelle to cell to tissue to organ to body to population. And this type of computational biology I call structurally integrated. So the difference between systems biology and this type of modeling, which has become known in the sort of NIH world as multi-scale modeling, is really about how structure at different scale determines function, and how function it develops across physical scales in space and time. And so all of these approaches are complementary and necessary. And, in particular, they tend to meet right at the cell, right? So, if we can build systems biology models of cells, then we can use available and new computational algorithms for modeling from the sort of atomic to molecular to subcellular scale, and then take the whole cell networks and predict from what's happening at the tissue, organ, and body scale. So I want to use just a few simple examples to give you an idea about these different approaches and how we use them. And my lab happens to work on sort of the mechanics of the heart and heart disease. So those the examples I'll tell you are related to heart disease, but I think this is a much broader paradigm. So if I start with an example of multi-scale modeling, we became interested a while ago in a molecule called deoxy-ATP. Now, of course, that ATP is sort of a key biochemical source of energy. And of course, muscles, like the heart, need to do mechanical work, and they get that biochemical energy to do that mechanical work by hydrolyzing ATP. But it's also possible for cells to make a variant of ATP called 2-deoxy-ATP using an enzyme called ribonucleotide reductase that converts ADP to deoxy-ADP, which is actually required in DNA synthesis. Remember, DNA is a deoxyribose nucleic acid, and this is where the deoxy comes from, the deoxy-ADP. So it turns out, though, that muscle cells with a little bit of deoxy-ATP mixed in with the ATP can actually generate more force and do more work. And so much so that converting a little bit of ATP to deoxy-ATP has now been proposed and even tested experimentally as a therapeutic for heart failure, because it has some very particular advantages for improving the force generating capacity of the heart without creating other problems. Now, heart failure, it turns out most of the therapies that you use to make the heart work harder only make the heart failure worse, because the physiology, the body is already trying to turn on the throttle. So you turn up the throttle even more and you only make the heart even sicker. But going back to the energy source and applying a more efficient energy source in the form of a small amount of deoxy-ATP seems to be a very promising way of helping the heart contract harder by making it work more efficiently. And so we wanted to understand at a molecular level how this was happening, especially how it could happen with just really even just 2% of the ATP in the cell being deoxy-ATP. It didn't really make sense. So what we did is we started by doing molecular dynamics. Okay? And the molecular motor in the heart is myosin, and it does work by binding and dissociating from actin in something called the cross-bridge cycle. And the cross-bridge cycle involves the hydrolysis of ATP. So essentially one ATP gets hydrolyzed for each cycle of the actin-myosin cross-bridge cycle, of the myosin attaching to the actin, generating force, detaching, and repeating the cycle. So what we did is we did molecular dynamics simulations of myosin with ADP because in the bound state of myosin attached to actin, actually the hydrolysis takes place. And so we actually modeled a myosin ADP bound to actin and compared it with myosin deoxy ADP bound to actin and solved for trajectories using molecular dynamics. And then once we had the most common trajectories, we did molecular electrostatics. And so you can see here, these are the charge distributions on actin, myosin ADP, and myosin deoxy ADP. And you can see, if you look closely, that the effective DAD of deoxy ATP or deoxy ADP bound to myosin actually changes the charge distribution on the binding surface of myosin to actin in such a way that actually increases the attraction, the electrostatic attraction between myosin and actin, which could then increase the rate of attachment and then increase the force development. And I want to go back because there was a little, we could then can then do Brownian dynamic simulation that you see there in the video. Okay, and this is now rigid body simulations of the molecule, and you can see the myosin is trying to bind to the actin. And if you do that thousands and thousands of times with both the myosin ADP structure and the myosin deoxy ADP structure, you can work out how does the on rate of the attachment rate of myosin to actin get affected by the presence of deoxy ATP. And the short answer was it increases it about 5-fold. So now we knew something very specific about the molecular action of deoxy ADP on the molecular motor. But there's much more to it than just the myosin binding to actin. It has to go through the whole cross-bridge cycle that you can see there. And in order for it to do that, this complex here, this little football-shaped thing on the thin filament that you see here, this is the thin filament, the actin filament, that contains troponin, and the blue rope there is tropomyosin. And calcium has to bind to troponin C. You can see the calcium coming in, a little yellow ball coming into troponin C, causing the troponin to rotate. It actually, troponin I dissociates from the actin, then allows it to rotate, and that causes the tropomyosin to shift. And as the tropomyosin shifts, it allows the myosin to bind. Okay? And then the myosin hydrolyzes the ATP, which is the green sphere, and releases the inorganic phosphate, generates force through the cross-bridge cycle, and then detaches. And so we have to make a model. If we want to generate, to predict the force development by the heart, we have to model all of these processes. And part of it is being able to model this process of the tropomyosin shifting, which you can see in this other video. And so with this information, we can start to build a state transition model that has a blocked state from tropomyosin, where tropomyosin is blocking the attachment of myosin to actin. Okay? And then it goes to a new state, B1, when calcium binds to it. So there's a calcium bound state. Okay? And then it goes to a new state, which is called the closed state. Okay? Now in the closed state, after calcium is bound, now the cross-bridge can attach. And so now a cross-bridge attaches, and we get a myosin actin state. And it turns out that that state further pushes the tropomyosin out of way and makes it more likely that neighboring regulatory sites on a thin filament can have a myosin bind as well. Then we go through the power stroke. So this is the part of the cycle where you can see the head, the myosin head rotating. And that's the part where mechanical work is being done, and the biochemical energy of ATP is being converted into mechanical work. And so now we have, this is also myosin actin. It's called the strongly bound state. And then you get release, okay? And so this is attachment, and then this is detachment. And we can use these simulations to work out these transitions between these different states and take the results of the molecular model and turn them into what we would call a Markov state model or a state transition model. And the thing that makes this complicated isn't this little diagram I just drew, because that's really just a few differential equations, one for each of these states. Okay? That's not the hard part. The hard part is the fact that when one tropomyosin, the tropomyosins overlap a little bit. And so when one tropomyosin moves, it tends to increase the chances that its neighbor will move. So imagine you're all sitting down. If we were in a classroom, you're all sitting down side by side and you're all locked arms. If the people on either side of you wanted to stand up, it would be energetically favorable for you to stand up. And if they wanted to sit down, it'd be energetically favorable for you to sit down. So this nearest neighbor interaction, this overlap of the tropomyosin causing this nearest neighbor interaction, results in what's called cooperativity. In other words, you get more of a sort of an all or none type of response. As some go on, all go on. Okay? And this explains why muscle has this very all or none type of response, very steeply cooperative activation. You go from a small amount of calcium to just a bit more calcium. You go from fully off to fully on in the force generating apparatus. So we use this type of model, this type of Markov state model, as the transition between the molecular models and the cellular model. And then we could use the cellular model to predict the effects of deoxy-ATP. Now here, R1, R2 represents the ribonucleotide reductase. So this is actually exogenous expression of the ribonucleotide reductase gene. And what it shows is that the muscle, this is actually showing the shortening of the muscle during a twitch, that increasing the upregulating ribonucleotide reductase in muscle cells causes an increase in the magnitude of shortening without significantly changing the amplitude of the calcium transient that's driving it. So it's a process that's autonomous to the cross-bridge apparatus, not the degree of activation. And here are the model simulations for different concentrations of deoxy-ATP. And you can see, even with very small concentration of deoxy-ATP, we're getting significant increases in the magnitude of the twitch force. And what we're not seeing yet is the fact that the twitch force could accelerate its relaxation as well with deoxy-ATP. You notice here that the red line, even though it's taller, actually comes back to rest faster than the green line. So the reason for that is because there actually is an impact on the calcium transient. It's not the magnitude, but the rate of decline. You'll see the rate of decline is faster when you overexpress the R1, R2 enzyme, when you have some deoxy-ATP, than when you don't. And so that's because there are ATPs doing a bunch of other things in the cell. And one of the things it's doing is running a pump. And that pump is a calcium pump that resequesters calcium after it's been released to activate contraction. And that's what causes the relaxation, the calcium going back down. Calcium goes up, triggers the contraction, the calcium goes down, triggers the relaxation. And if that pump, which depends on ATP, can go faster, then actually the force the force falls faster, which can be very helpful. Okay? And so that is a whole separate project that I won't show you, where we now are also doing the same sort of combination of molecular models and Markov state models and cellular models to predict how deoxy-ATP affects this calcium pump in the cell. Okay? So that's just an example of how we can sort of go from molecule to cell to understand, in this case, a putative therapeutic mechanism. How do we go from cell to tissue and organ? Well, so you could think of this model here as the whole cell model, this as being a cartoon for the Markov state model, in this case of an ion channel. Okay? And so we do this in the case of the electrical and calcium handling activity of the heart by modeling each ion channel as a Markov state model, and then putting all those together into a whole cell model to model the change in voltage across the cell during the action potential. Then we couple the cells together in a model that has the electrical coupling conductivity from cell to cell, so the action potential can propagate. And then it propagates primarily along the direction of the muscle fibers in the heart. So we need a model of the whole heart that has the muscle fiber architecture in it. We determined by diffusion tensor magnetic resonance imaging. And then we solve the three-dimensional equations that determine the spread of that action potential impulse in 3 dimensions to predict the electrical behavior of the whole, in this case of the left and right ventricles of the heart, as the impulse spreads. So in other words, there's a paradigm for going from protein to cell to tissue to organ that already exists, at least in the case of the propagation of the action potential in the heart. Now, it turns out there's also a paradigm for the mechanics. So we can do the same thing. We can take the kind of Markov state models of myofilament interactions that we derive from molecular models, like I told you, and then we can take the forces generated by each cross bridge and distribute them in 3D through the hexagonal lattice of myofilaments in the cell. We can then take those stresses developed by the cell, and using biomechanics, using engineering mechanics, we can simulate how those stresses distribute themselves in 3 dimensions through the tissue, knowing its 3-dimensional structure. Then we need the boundary conditions, which are actually the resistances and impedances imposed by the vasculature on the heart, and the volumes or upfilling pressures from the venous side back into the heart. So using a model of the circulation, we can generate the boundary conditions on the heart, and then putting all that together, we can predict the 3-dimensional mechanics of the heart. So this is a sort of an engineering paradigm for going from ordinary differential equations or Markov state models at a point representing a protein or an ion channel or a complex to a 3-dimensional model representing the whole organ as a continuum in which the electrical activity and mechanical activity are distributed continuously throughout them. So feel free to stop me if you have any questions. So if you can make an electrical model of the heart and a mechanical model of the heart, maybe you can make an electromechanical model. And we thought about doing this in the context of a condition called dysynchronous heart failure. So it turns out that a lot of people with heart failure also have some electrical conduction defect. It's usually caused by something called bundle branch block, where the sort of the highways that propagate the electrical impulse to the left and right ventricles get blocked. And that slows down the spread of the impulse through the heart, and that makes the contraction less synchronous and less efficient. So this dysynchronous heart failure is a weak contraction of the heart made worse by an electrical conduction disorder. It's quite common. And it turns out, though, because of this, if you could resynchronize the electrical activity of the heart with pacemaker technology, you can actually improve the mechanics. And so this is an unusual therapy where you actually use an electrical therapy to improve mechanical performance, developed about 20 years ago, and actually it can work very well and not. And so here's an example of 8 patients at the VA, and you see just how it works. You can see in this patient here, this lucky patient actually improved his heart function by nearly 60% after having this therapy. And it happens almost immediately. They wait a few months to do the measurement, but as soon as you turn the pacemaker on, you get an immediate improvement. In the best cases, you get a continuum. That improvement in turn promotes a further reversal of the disease. And that's clearly what happened in this patient. Then you can see there were 3 other patients who got between 12 and 16% improvements, and anything over 10 is considered really good. 0 and 10 is kind of borderline, but a 10% improvement was worthwhile. These patients in often cases are very sick. Now you can see that there were 3 patients here who actually got less than a 10% improvement or actually continued to get worse. Interestingly, they actually felt better. So whether this is a placebo effect or whether this is an effect that sort of doesn't reach the stage of being able to be measured by echocardiography, but still results in some improvement in perfusion, is less clear. So these would be called echocardiographic non-responders. They didn't actually respond by an objective measurement, but they're called clinical responders because we asked them, they said they got better. Okay, and then here's the one clinical non-responder, and you can see that that patient continued to get worse. So there's a huge variation. So this is the thing. It works, but it only works half the time. Maybe if you're more generous, 3/4 of the time. Okay? But there are some people it just doesn't work at all. So it'd be really helpful to know either how you could change it to make it work better, or who it wasn't going to work in, so that you weren't going to spend $100,000 for a procedure that didn't work. So we decided that we could make patient-specific computational models of their electromechanics to see if we could learn anything that might help us predict who would respond and who wouldn't, and how we could make the therapy better. So first we used CT scanning or occasionally MRI to reconstruct anatomic models of ventricles. Then we did electrocardiographic measurements by a variety of different techniques. Mostly we settled on something called vector cardiography, and use that information together with nuclear medicine imaging called SPECT. So this is a kind of imaging that uses a radioisotope tracer to see if there are regions of the heart like this one that are ischemic, that have a lack of blood flow, because many of these patients also suffer from coronary disease. And about half of them did. So now that we have this information, the electrical information, the anatomic information, we can actually put that together to make an patient-specific electrical model of that patient, left and right ventricles. And you can see this patient had left bundle branch block, which means their impulse started early on the right side and ended late on the left side instead of being more synchronous. Next, they went to the cardiac cath lab where we measured pressures and did echocardiography. And we could use that to make a mechanical model that we could couple to the electrical model to make a coupled electromechanical model. So what you see there is the electrical activity superimposed on the mechanical activity. And then we could actually simulate the effect of the therapy itself. So here's the patient with left bundle branch block. And you can see the impulse starts on this side and ends on this side. Interestingly, after the pacing, it actually was the other way around in this patient. It started on this side and ended on this side. It didn't actually look any more synchronous after the therapy than before, but this was the therapy that the doctors thought was most optimal. And interestingly, this patient got better, even though the overall dysynchrony wasn't necessarily improved. And so this is maybe a clue that the goal of trying to actually synchronize the electrical activity is not maybe the goal. They thought it was the goal, but when we showed them they hadn't synchronized the electrical activity very well, I thought, well, maybe it's something else we're doing. And so one of the main hypotheses is that this therapy works by increasing mechanical efficiency. So how much oxygen consumption and ATP and work of the muscle are converted into work of the heart, output of the heart. And clearly, if you have the two chambers working dysynchronously, you're going to impair the efficiency of contraction. So what the model showed is, in fact, the pacing did, in fact, improve the mechanical efficiency of the heart. But the interesting thing is that it actually improved it in both the responders, the 4 patients who did well, and the 4 patients who didn't do so well. And interestingly, the improvement was actually only statistically significant in the patients who didn't do well. Okay, so how's that? So yes, cardiac resynchronization therapy does improve mechanical contraction, but guess what? Improving mechanical contraction doesn't actually make you get better. If anything, it may actually mean you're more likely to not get better, which is super weird. Okay, so we ended up, sending us on a long investigation to, if not efficiency, what? And we did find something, and I won't elaborate on exactly what this marker was, but it was a regional measure. It had to do with a particular part of the heart wall that's particularly vulnerable. And it turned out if we focused in on that part of the heart wall, the septum between the 2 chambers, that's where we found predictors from the model that not only correlated very well with the outcomes, but also was significantly different. See, the responders started high and went down a lot. The non-responders were already much lower and didn't improve much. So we now have a new tool for helping to predict which patients will respond best to this therapy, and maybe to optimize the therapy so we get more of them into this range. And there's a new company that's commercialized some of this technology over the past 2 years. So maybe I'll stop there and see if there are any questions. No? Okay, well, let me talk next about, the real thing about a therapy like this is, it's nice that you turn the pacemaker on and instantly there's an improvement in function. But actually what's really important, and you can see that in the case of our best patient, is that actually you really want to reverse the adverse changes in the heart that have led to the heart failure in the first place. And that takes time, okay? And that process is called remodeling, okay? And so the adverse process is called remodeling, and it generally is a reflection of the fact that the heart changes size and shape as it gets sick. And what we would like to do is promote reverse remodeling. And in our best patients with undergoing cardiac resynchronization therapy, what happened is they experienced reverse remodeling. So the adverse changes in the size and shape and function of the heart that occurred with the disease actually reversed. And that's really the holy grail for the therapy and treatment of heart failure. So there's two main ways that the heart remodels in response to changes in its environment. One is by dilating, getting larger, and this tends to happen in these patients with who need CRT. And that dilation is caused by the cells getting longer. It's called eccentric hypertrophy. The other type is when the walls thicken. So instead of the diameter getting bigger, the thickness increases. And that's due to the cells getting fatter. It's called concentric hypertrophy. Okay. This tends to be associated with a volume overload, an increase in the filling pressure on the heart. This tends to be associated with a pressure overload, an increase in the arterial pressure acting on that the heart is beating against. And often these two things happen together. One will lead then to the other, and you'll get a heart that's both dilated and thickened, so just a massive heart. Okay. So for a long time, people have wondered, well, what's the difference? Why does one type of hemodynamic load cause one kind of remodeling, the cells get longer, and another causes another type? And so this caused us to come up with a hypothesis that was based on some experiments we'd done, where we said that if you stretch the cells along their axis, they'd want to get longer. And if you stretch them primarily perpendicular to their axis, they'd want to get fatter. It almost sounds ridiculously simple. Okay? And we would not have invented this on our own, but we did some experiments and this seems to actually happen. And so then we put it into a computer model to see if you just say these two simple rules. If the maximum stretch along the cells increases, will the cells elongate? And if the maximum stretch perpendicular to the cells increases, will they fatten? And the simple short answer is when we put those rules into a model, this is what happened over a period of a month or so. In the model where we increased the filling pressure, it dilated like eccentric hypertrophy. In the model where we increased the resistance, the arterial pressure, we got concentric hypertrophy and thickening. So something about this really, really simple-minded idea seems to actually work. But this is just a purely empirical idea. There's no biology here, right? So this brings me to the last part of the talk where we talk about systems biology. We know that cardiac cells, both the fibroblast that makes the extracellular matrix and the myocytes that do contraction, are strongly sensitive to mechanical signals. They're sensitive to stretch, and they're sensitive to the stiffness of the extracellular matrix. And the myocytes, as we've heard, tend to respond by hypertrophy, and the fibroblasts tend to respond by adding more extracellular matrix, which can be a bad thing. It's called fibrosis. And together, this is called tissue remodeling. So we want to understand the mechanisms by which abnormal loads cause abnormal hypertrophy, abnormal growth of the heart cells, and abnormal fibrosis, abnormal accumulation of extracellular matrix in the disease tissue remodeling process. And so, to cut a long story short, what we did is create this network model from the literature, either in cells that have been stretched or in hearts that had been hemodynamically overloaded. And you can see that this, and the driver of all of this is a mechanical loading or stretch. And this network came entirely from the literature, and you can see it has pathways for calcium signaling, MAP kinases, cytoskeletal signaling, PI3 kinase AKT signaling. These at the top of the receptors, these are transcription factors, and these are genes. All together in this model, we had 11 transcription factors. And we only show 14 of them here, but there are actually 784 target genes, about 300 of which have been experimentally validated, the rest were predictions, 921 reactions, 14 translation reactions. So it turns out 14 of the genes were actually coding for proteins that were in the network itself. So we fed them back into the network. And this all came from 115 published papers. So we made this network model, and we implemented this network model as a system of ordinary differential equations. And in case you're wondering, with a model that massive, I must be able to make anything work, right? As von Neumann was said to have, was quoted by Fermi as saying, with 4 parameters, I can fit an elephant, and with 5, I can make him wiggle his trunk. Well, I don't know that's really true, but oddly enough, a group in Germany did actually figure out with 4 parameters, or complex numbers, so I'd say there were 8, they actually did make an elephant. And with 5 parameters, they even made it wiggle its trunk. So maybe von Neumann was right, and maybe this model is a complete waste of time. The problem is, this is not an elephant. This is an elephant. And I know because I was there and I took this picture in Africa. And that's what we have to deal with. We're dealing with an elephant, and so we need a lot of information. We need to get up close and personal with it. And the way we avoid this model from being over-parameterized is we actually use the same parameters for every class of reaction in the model. Okay. So there's only a handful of parameters, free parameters in this model, and as many of them as possible were determined just from empirical data. So there was, by the time we were done, there was no parameter optimization, no fitting here. But we do have to be able to test the predictions. And so what we do is we make these micropattern membranes that are very thin, and we grow neonatal cardiac muscle cells on them, or human stem cell derived myocytes on them. And these grooves make them take on this sort of lifelike, adult-like, in vivo-like, elongated, striated phenotype. Okay? And then we put them in a stretcher such that we can stretch them primarily along their axis or primarily transverse. And then we did RNA-seq. And here are the main clusters. I won't take you through them in detail. These are Go biological processes, gene ontology, molecular functions, cellular components, and KEGG pathways. So very classical type of stuff. I'll just show you how many of these same pathways were also enriched in the model. All of these ones. These two, these ones, and these pathways. So there are pathways that are missing. There are genes that are missing. Interestingly, they tend to be the downregulated ones. Okay. So there's more to add to the model, but we do get quite a bit. Okay, so what can we do? Well, here I'll just show you one thing, which is the p-value for the genes in the RNA-seq. So, numbers on this is the log base 10. So numbers on this side are highly significantly upregulated or highly significantly downregulated. Ones in the middle are not significant. And this is the fold change predicted by the model. And you can see, actually, if you compare these different counts of genes in the different categories, the model actually overall is about 77% accurate. So it correctly predicted that the genes went up, down, or stayed the same 77% of the time. Okay? And 23% of the time it didn't. And so these errors, we've actually been able to now look at the errors and find ways to, what was missing from our model, what possible mistakes were there. And so this has become our way of sort of iterating the biology. Is we look at what the model can do, then we know that the pathways can explain that, and now we really focus on what it can't do. Okay, so with that, I'm just going to close and say thank you, and hopefully leave some time for questions or discussion. You're as shy as the students in my class. It's okay, no dumb questions.
If you have any questions, just feel free to unmute yourself or type in the chat.
If you're interested in this kind of systems biology, we're now recruiting students to, including undergrads, to work on making these kind of models for a whole range of different problems and phenotypes. So not only in the heart, but in the pulmonary vasculature, and in skeletal muscle, and not only under disease conditions, but also under conditions of health. So we're actually really interested in what kind of loading conditions during exercise promote positive remodeling. And so we have a whole new project on modeling human performance. So lots of research opportunities to apply systems biology, not only to disease, but to health.
I guess I have a more general question about like bioinformatics in general. I was wondering what you think the difference between, say, computational biology is and bioinformatics. To me, I think bioinformatics is sort of like a subset of computational biology. So I was just wondering what your thoughts on that was.
Yes, yeah, I think bioinformatics is a subset of computational biology. And I mean, I do think there's sort of emerging areas of computational biology that are sort of integrative in a fourth way, and that's the sort of knowledge integration through machine learning. Okay, but I would say the way I would see machine learning and data sciences is sort of infiltrating all of these different areas. So we use machine learning and data science across all of these different strategies, modeling strategies. I think the key difference is that it's sort of more on those lines I drew at the interface between the experiment and the numerical analysis. Where the machine learning and data sciences really come to the fore. But I think if you took those four areas, information integration, functional integration, structural integration, and data science, you'd be hard pressed to find that, first of all, any of them weren't integrative, and secondly, that there was a branch of computational biology that didn't fall under one of them. So I think it's probably a good sort of working model of computational biology.
So does your lab use machine learning and data science in your modeling of cardiac systems at all? Yes.
So we use it in a number of ways. So one way in the multi-scale modeling world, I didn't show you, but we get a lot of data from patients, electrical data and anatomic data in particular. Machine learning is particularly valuable for analyzing imaging data. So we get cardiac MRIs from patients with congenital heart disease, sometimes children, sometimes adults. And cardiac MRI is not a common procedure in most heart disease patients, but it is common in congenital heart disease patients. And so lots and lots of congenital heart disease patients at Rady Children's Hospital get MRIs every few years or more often. And what we do is we use unsupervised machine learning techniques called statistical shape modeling to make unsupervised models mostly using principal component analysis, and we get the major modes of shape and function variation in these patients that we can then feed into supervised machine learning algorithms to discover features of these cardiac images that are then predictors of clinical outcomes or early markers of disease. So that's one big area, dealing with clinical data. The other is when we're dealing with genome scale data. We can use machine learning strategies in order to discover new networks from existing data sets, and I'm sure you all learn a lot about those type of methods. And so while I described a model that was built from the literature, you can now start to fill in that model with new pathways that you learn by applying machine learning algorithms to genome scale data sets like transcriptomic and proteomic data sets. So that's the third way we use machine learning, I guess, is we actually use computational models like the ones I showed you to train machine learning algorithms so that we could, instead of actually making a model of the patient, we could use the models to predict what the features are, but then just by training a machine learning algorithm with thousands and thousands of models, we could just learn directly from the data that went into the model what it was about the data that predicted the outcome. So in the end, we can use machine learning to put ourselves out of business, or at least to make it so that when you go to the clinic, you don't actually need to execute a whole model. You can just execute a machine learning algorithm that's been trained by running models hundreds of thousands of times.
That sounds really interesting. Does anyone else have any questions? Feel free to put it in the chat.
Hi, I have a kind of a question, just general one. Nice presentation, by the way. It was really interesting. Thank you for sharing with us. My question kind of goes off of what you said when you showed the graph of the elephant and said, well, this is not actually an elephant. So in this sort of really model heavy work, I'm assuming that there are differences when you have actual people and how they respond versus the model's predictions. When that happens, how does your lab respond to those disparities?
Yeah, that's a great question, Joelle. This is both, I think, for us, the biggest challenge and the biggest opportunity of the patient-specific modeling, right? So one way is you can make a model of each individual patient and use that model as a framework for putting as much different patient-specific data in as possible. And so in that sense, the model becomes a tool for data assimilation and data fusion. The other is to use, and that's nice, but it's very time consuming and you don't know that you're getting all the right information, right? Maybe part of their measurement that you got in the clinic had to do with what they had for breakfast this morning, right? Or how much sleep they got. And so by not being able to account for that, they're going to have variations that you can't explain. The other strategy we're using, like I said, is these sort of unsupervised machine learning methods that what they basically do is they give us sort of statistical representations of variations in a lot of different features across the population. And they allow us to rank them and find out what are the most important, right? And so given that you can never account for every patient-specific difference, this sort of unsupervised machine learning approach allows us to find out what are the most important differences and account for that. But this is a constant consideration, and particularly in the difference between dealing with animal models where all the animals are very similar and humans where every human is different. This is kind of, I think, a key question. It'll never go away, and it's the foundation of what's behind precision medicine. The idea is if you measure enough and you synthesize it properly and you focus on the right things, you'll do a better job. But we'll never be able to sort of predict at birth, every disease that someone could have, because a lot of them are affected by things that are not, in our genomes, that are dependent on our environment and the whole history of our life events.
Yes, that makes a lot of sense. Yeah, thank you. Another question off of that, for your patient-specific modeling, since that seems very specific, it's in the name, how it seems like a lot of when we're trying to make bigger conclusions and it's very different per person, how like it seems like might be like a lot of money or a lot of just something for one person to make an overall conclusion. How does that kind of work?
Yeah, I mean, that you're right. The difference, though, I think, for say, I think it has a lot to do with what the therapy is. For example, if the therapy was a drug, then you'd have to have a lot of different drugs to make patient-specific models, right? To be able to then provide a patient-specific therapy, right? I mean, in the ultimate, you could make a patient-specific model, but you'd have to make a patient-specific drug, and it's never going to happen, right? So unless you have a wide variety of different drugs that you could simulate, there's a limit to what you could learn from the patient-specific model. With devices, it's a little different, right? Because the devices can be programmed and implanted in different places. And so there's a lot of different ways you can deploy the device. Plus, device is a very specific thing, and it's very expensive, right? Probably the doctor just say, well, try this drug. And if it doesn't work, we'll try another drug, right? But you can't say, well, we'll try this surgery. And if it doesn't work, we'll try another surgery, or we'll try this $100,000 device, and if it doesn't work, we'll take it out and put in another $100,000 device. So I do think that it depends a lot on what you're going to do with the information, but I think something like a very expensive, invasive, complex device therapy like cardiac resynchronization therapy is probably a better target for this type of highly patient-specific modeling than, say, a pharmacotherapy. Now, maybe it could be true for a very customized, very expensive chemotherapy, for example. So I think you're right that there are limits to how it generalizes, which is why I also like this sort of complementary population-based approach we're using, where you can learn things about whole populations and come up with, use machine learning to discover and models to discover new markers and say, okay, you're at higher risk, so let's look at what your risk factors are.
All right, that makes a lot of sense. Thank you so much.
Thank you. Well, it's super nice to talk to you all. I hope you're going to have a good spring quarter, and it'll be the last time you have to be stuck behind Zoom if you don't want to be. And you can be back in classes in the fall and the summer. And certainly look forward to seeing you. I hope by the summer even we'll be able to have students come back, undergrads come back into the lab.
Thank you, Professor McCulloch. It's a great talk.
Thank you. Bye-bye.